Hello everyone, welcome back to Chinmoy's Biology channel, and today we are going to discuss about the linkage diagram in NCERT book. So that is a very important diagram, but it is very difficult to understand. So today in details we will study only that, and as in the previous episode we have already spoken about a bit of linkage, recombination and crossing over. So if you have not gone through that, I'll be putting the link down here. So let's get started with the important diagram of linkage. So here we are now with the important diagram of this linkage, right? So what they are showing here, they are first showing us that cross A and cross B. So what does this two mean? So please concentrate on this diagram full so we will understand it in a very easy manner. So cross A means first it was done with these set of organisms, and then in cross B it was done in the second part. So cross A was done first. So this is the symbol for female and this is the symbol for male. So here these are the parental chromosomes. So this is Y is yellow and W is white, and this is the yellow white chromosome, and this is wild type. So what is wild type? Wild type we are denoting with Y plus and W plus. So wild type is always the dominant type, and it is the normal one which is not produced in the lab. It is the normal type of chromosome. And this, what is this type? This is actually the type which is being synthesized in the lab. So Thomas Morgan, he did these experiments in lab. So in a synthetic material he did this crossing and all. So this all were the recessive type, and which he found in his experimented and produced in his lab. Is it clear? Now this was the parental generation. So this is the female one having 2 X chromosome, and here in the male one is X, and this different type of purple chromosome is the Y chromosome. It is clear to all of you now, right? Now next we are moving on to F1 generation. So when we are moving on to F1 generation, we can see here that this Y and W, these two genes are located quite close to each other, both in this yellow white as well as in this yellow and white wild type. So they are having a great linkage bond. So in F1 generation, what we are seeing, that they are exchanging their places. So YW is having this chromosome here. So they are exchanging their places, and this YW has come here. But they have not broken that pair. So they have come in pair itself. Okay. So this is why this has become wild type now, because the dominant has come here in pairs. And this has become the recessive now, wild and yellow and white. Sorry. So yellow and white. Is it clear? Now in the next F2 generation, what we are seeing, in the F2 generation, if we concentrate, that is parental generation, they are telling there are 98.7 types of parental offsprings. They are looking like similar to parents, which are carrying the parental genes. And only 1.3% are having recombinant genes. Okay, that is a new form of gene. Okay, so let us see here what is happening. Now the first 4 they have given, they are male. So the first 4 are male. So you can find out why they are male. They all are having this Y chromosome. And the next 4 he has given of female. So they are all having this X chromosome only. Now, what has happened here in the F1 generation in cross A? You can see here, this first male, it is wild type only. Here Y, these left side are all parental. Okay, this is Y and W, that is wild type. They have come together and it is dominant wild type. Here yellow and white, they are together. They are like recessive. Now here what has happened here, you can see that they have broken and combined. So Y plus has come with this normal W, and here Y has come with W plus. So here white has become recessive and here yellow has become recessive. So there is a recombination here where part of genetic materials of the chromosome has been shifted here. So the full chromosome has not gone as it is. A part of the chromosome has shifted from one chromosome to another. So that is what we have studied in the previous episode. That is, a crossing over has happened to form a recombinant type of daughter cells. Okay, or we are seeing here that the next generation of offsprings are having recombinant types as well. So it's clear to all of you. Now we will move on to the female of this generation. Here also in left side we are seeing the parental types, and in the right side we are seeing the recombinant type. So what is happening here? Here same, from the parents this full chromosome has come like this, and from another parent this YW has come. And in the second also both the YW has come here. Okay, it's fine. Now again, two sets are there where recombination of this Y plus with W and Y with W plus has happened as same as the previous one. Okay, is it clear? So this only 1.3 part is recombinant. That is very rare. So that what happened in cross A. So is it clear to all of you now about cross A? Now we will move on to this cross B. So what is happening in this cross B? Now here when we come to this cross B, same thing is here. This is the female and this is the male. But the first difference what we see here, you can see here the genes are located quite near to each other. So we have spoken in the previous episode also that the nearer the genes are located on the chromosomes, the linkage will be far more. But here you can see the genes are located farther away. So the distance between the genes is far. So the linkage in the genes will be lesser. So there is a tendency of more and more recombination in these cases. So here what we are seeing, the females are white and miniature. So what does this miniature means? That they are mini, they are small, they are having small wings and small bodies. So they are miniature in nature. Now here the same with the wild type, they are having W plus and M plus written on that genes, that they are the dominant part. Now in the F1 generation, the female is coming as W and M and this W plus and M plus. So this has come in full from the parental generation. Now here the full this W and M has come here. So that has happened, but the genes have not mixed in the F1 generation. Okay. So what all gametes have been formed in the F2 generation? Same as here, the first thing has been the male part, as they are having the Y chromosome here you can see, and the next part is the female part as they are having only X chromosomes. Okay, so what has happened here, you see. Now here the parental type, they are telling it is 62.8%. Okay, so here we have spoken it is 98.7%. So mostly they were of looking like parental, but here as the linkage was less, only parental type was 62% and 37% were recombinant. So they have recombined their genetic materials. Okay, so there is a recombination. More crossing over has happened in this cross B. And these all are variations from Mendel. He told that the genes or the alleles, they are independently assorting. In his law of independent assortment, he has told that they are not mingling or they are not interfering with each other. But here, there is a complete deviation from that. We are proving that there is a recombination, there is a crossing over, and they are not independently assorting. Okay, so let's move on to this one first. Now here you can see in this first males what has happened, W plus for M plus in the left side, they are all like parental, and the same like this W and M, it was Y and M here. Now again in the recombinant, the W plus has come with the M and miniature, so it has formed a miniature one. So it is a recessive again. It has become white. We can see as white has become this dominant, and as white has become recessive and miniature has become they have differentiated. So when both were together, they were dominant, and when they are separated, when they are coming independently, then the recessive part, which was here miniature and here which is white, which will be shown or represented. Okay, so again in the next part, the left side is parental, that is W plus and M plus, the same chromosome with the same genes are coming here from the parents to the progeny. And here also the same thing has happened, that it has become the wild type because this wild type is dominant here. Again here white and miniature will be here, as all the alleles are white and miniature. So all recessive genes have come here. And we all know that this is a homologous chromosome. So as it is homologous, no mixture is there. White and miniature will be the genotype here. Now next comes the miniature here in the recombination, W plus M and W and M. So it has become miniature and has become white. So W and M and W and M. So this mixture, this recombination, this crossing over has happened. So these are the results of 2 dihybrid crosses conducted by Morgan. Cross A shows the crossing between genes Y and W, we have taken here, and cross B showing genes W and M. Okay, so here the dominant wild type alleles are considered in the plus sign, we have told you earlier. So this is all about this diagram only, which is very difficult to understand, but once you understand this diagram of linkage in your NCERT book, just go and check once. If you are not understanding, then also please rewind my video from the beginning, start once again, read, listen to this carefully. Still, if you are having any doubts, write in my comment box. I'll get back to you again and again to teach you this concept. So if you are understanding this concept, it will be very easier for you in the next classes to understand the molecular basis of inheritance and all. It will be very easy. So in the next class, I'll be teaching you that why Drosophila only was taken as the experimental animal. So what are the benefits of Drosophila? So as we know that why Mendel chose the pea plant for his experiments, like that only we should know why Morgan chose Drosophila for his experiment. So what are the importance of that Drosophila? We will read that in the next class. So for that you need to stay tuned, subscribe to my channel, share it with your friends, and if you are liking my channel, please do share it as much as you can and subscribe to my channel. Stay tuned, stay updated. Thank you. Bye bye.